Let me pose a question. What links muscles, enzymes and snake venom? Well, judging by the title of the video, you probably could guess, but over the next two videos you'll find exactly how to link those three topics together. This will be probably the most recognisable polymer out of any that you've seen. The one that you think of as soon as you think of polymer. But as we learnt in the first video, in order to be a polymer you have to first have a set of monomers. Now for proteins, each of these individual sections are called amino acids. There are 20 and they look like this. Remember the R group from the last video? Well, we'll use that a lot more often here as well because each of these 20 amino acids have different chemical formations, and to remember them all is a bit tedious and kind of unnecessary. But something else you should remember is that C double bond O OH, the Cu group. If you remember in the previous video this was called a carboxyl group. Now the carboxyl group gives it its acid part of the name, amino acid. So what does the amino mean? Well, if you look on the other side of the molecule, you see the NH2. That NH area usually corresponds to an amino group. Now, can you guess whether this is acid or alkali? On one side, you have the acid, but on the other side, this NH2, if you know quite a bit about chemistry, you'd know that ammonia acts as a weak base. So this molecule has both acidic and basic properties. So on one hand, if it's put into a system that has a high pH, so very alkali, the acid part will react. And on the other hand, if it's put into a very low pH, so very acidic, the basic side will react. This molecule is called amphoteric. Let me ask a really difficult question. What reaction do you think will form one of these? Wow! Condensation. Yeah, as I said before, most of these molecules are formed by condensation and broken up by hydrolysis. It's kind of an easy thing to remember at this point, and it's kind of a repetitive point, but nevertheless, it's still formed by condensation reactions. And these condensation reactions take place between two of these molecules to form a longer chain. In the same way that with saccharides, we had two of them joined together to form a disaccharide. With amino acids, when two of them form together, you get a dipeptide, and that bond between them is called a peptide bond. And I suppose it's almost important to note that these R groups are unaffected by this change. Now, what if we get a long chain of these? Well, just as the case with saccharides, we have a poly of this molecule, which in this case is a polypeptide, or in a more common everyday term, protein. And it's not uncommon to see these amino acids bond to different amino acids. There are 20 of them that can bond in any formation to form a protein structure. Interestingly, these protein structures are usually coded for by the animal's DNA where they're formed within, which we'll learn later on. There are 4 different types of structures of proteins. The first one being a primary structure. Very simply, the primary structure is where these amino acids create a long chain, and the primary structure refers to this chain itself and how this chain is composed. The second part of the structure is secondary. So in this situation you have 2 forms of secondary that you need to remember. First one is alpha helix. This is where the proteins spin in a spiral. And the second kind is a beta-pleated sheet. So this secondary structure is usually formed by hydrogen bonds in the carbonyl groups interacting with the hydrogen bonds on the other side of the structure with the amide groups. It tends to be hydrogen bonds that cause this secondary structure. The bond part isn't entirely accurate, it's more of an intermolecular force, but it's the strongest intermolecular force that can be formed between two existing molecules, and that happens when there's a slight, what's called a dipole, where the molecule acts as an almost magnet with two sides, one that's slightly more positive, one that's slightly more negative. Between those two molecules, you'll have an interaction. Depending on how the hydrogen bonding presents itself, the structure will either be this spiral or this beta-pleated sheet. Now, there's no way you're going to get the third one. It's a tertiary structure. The names are quite logical, thankfully, so we don't have to learn someone's name for a structure of protein. These tertiary structures are formed by the bendings and inner workings of the alpha and beta molecules that we've discussed earlier. However, not only hydrogen bonding is present, but also things called disulfate bridges, which are bonds formed by sulfur atoms in a covalent bond. These are a lot stronger than the previously mentioned hydrogen bonds, and they physically bind to each other, causing the bends in the tertiary structure. Finally, quaternary structure. Now, this is where multiple sections of a tertiary structure will bind together to form one big molecule of protein. Things like this include the hemoglobin that I mentioned earlier, using the alpha helical structure, which then binds into a tertiary structure and is then added to 3 other structures to form hemoglobin. Of these 3 other that it binds to, one of them will be another alpha helical structure, and the other 2 are referred to as beta structures or beta chains. What's interesting is that iron is able to bind to these structures in 4 separate places due to the R groups of hemoglobin, which allows oxygen to carry through our body on things like blood cells. Ionic bonds can also form in these tertiary and quaternary structures to help keep the structure in this tight state. Ionic bonds are when the charge difference between 2 ions is so great that they're attracted to each other, which, like the disulfate bridges, but instead of being bonded together by electrons between them, they are drawn together by the charge difference between them. Now, finally, we need to know how to do a test for these structures, and this isn't as complicated as the lipid test, nor does it have as many steps as the disaccharide tests. This test is called the Buret's test. For this, again, we form a solution in which we put the sample we want to test into a test tube and dissolve it in water, but this time we put potassium hydroxide in, and we continue to stir up the solution for about 2 minutes to 5 minutes, depending on how thorough you want to be. But after this point, we put in the Buret solution, which, if there are any amino acids present, will turn into this purplish colour, purpley pinky colour, and that's the first bit of proteins. We'll do more again next time on the further structure of proteins and the structure of enzymes and how that all relates.